Hello and welcome to the talk on next generation cancer care in the age of genomics, precision medicine, and hybrid biology. My name is Jimmy Lin, and I'll be sort of talking through this. My background is um, my bioinformatics training was done at Yale, then did my MD PhD at Hopkins, and has done work at WashU, looking at implementing cancer genomics. And now I'm the founder and president of Rare Genomics Institute, sort of to talk a little bit about this. Um, I'm told to remind people that if people have questions, to feel free to put them in the Q&A section as we go along, and we will answer them at the end. Great. So in order to first talk about sort of cancer care and cancer genomics, we really have to really start at the beginning. What is cancer, and why is it that we're even thinking about genomics and cancer genomics? It first started off with a seminal paper, really in 1990 by Eric Fearon and Bert Vogelstein, looking at a genetic model for cancer tumorigenesis. And what the fundamental sort of finding in this paper that it outlined is that cancer ultimately is a genetic disease. This was actually a very fundamental and foundational understanding that cancer is not, some sort of infectious disease or something that's sort of other causes, but fundamentally it is a disease of genes. There are other things that can affect the genes, but ultimately it's the genes that's important. And that's why if the genes are important, the genomics will be important, and that's really starting off the field of cancer genomics. So historically, there has been understanding of cancer in terms of histopathology in the different states, starting off with sort of hyperproliferation to small polyps, to specifically looking at colorectal cancer, and into severe dysplasia, adenocarcinoma, and causing the cancer. And what this paper in 1990 really was able to connect is providing a genetic model for each of these steps. At each of these stages, then linking what's observed in terms of the histopathology to genetic changes, whether it be a loss or a gain of specific genome parts, or there be specific mutations, or even sort of methylation changes that will be affecting the genetic background. So this really outlines the connection between the genetics of cancer and what's observed in terms of the histopathology. One of the first important genes that was discovered through the work of Vogelstein and others was the discovery of p53, sequencing many human cancers and finding sort of a loss of 53 and defining for the first time p53 as a tumor suppressor gene, and laying the foundation for cancer genomics. Subsequent genes have been found, such as APC, which is shown here, or in terms of looking at EGFR and gliomas. So this really sort of started a whole revolution of finding genes that were genetically altered that has later been really important in our understanding of cancer. So subsequent to that, looking at, besides actually single genes, there was looking at larger families of genes, specifically, and this paper shown is the tyrosine kinome, which is the full complement of tyrosine kinases in colorectal cancers. And this published in 2003, finding that there were a large number of tyrosine kinases that were mutated at high frequencies in colorectal cancers. Subsequently, similar work was able to discover PIK3CA as a novel oncogene for cancer, not only finding the gene, but also finding significant hotspots in that. And of course, PIK3CA now is a gene of significant clinical interest, be able to develop therapeutics for it. And not only looking at tyrosine kinases, similarly looking at tyrosine phosphatases is another paper that was published subsequently in 2004 in Science, looking at these sort of gene families in cancer. So ultimately, this laid the foundation of specifically looking at the genetics of cancer, and that being very important. And starting out by single genes and looking at gene families. And of course, we know there's this big change in around 2001, 2003, and subsequently with the finishing of the human genome. For the first time now, we can not only look at single genes or gene families, but the entire complement of genes, whether it be the exome or the genome for cancer. So some of the first exome studies was done at Johns Hopkins, and part of our work there when I was a graduate student there, looking at the full complement of genes there. And this is sort of prior to the implementation of next generation sequencing technologies. So some of the early studies that were done there using mostly sort of Sanger technologies was be able to look at first of all, the consensus coding sequences, the CCDS genes, sort of 20,000 genes for breast and colorectal cancer, sequencing all their exons and be able to find the mutational profile for cancers for the first time. So this was initially published in 2007, looking at it and finding that, importantly, that the mutational profile of cancers was not specifically to one gene family, but it actually was an entire landscape of it. Subsequent work, we were able to actually formalize this and later that year to map it out, showing in the picture here that if you take a look at the landscape, which is shown if you take the chromosome line end to end from chromosome 1 all the way to the chromosome 23, the height of the landscape being the mutational profile of these cancers, that there are some cancers that are mutated, some genes that are mutated at high frequency, which are well-known cancer oncogenes or tumor suppressor genes, but a lot of these smaller genes are mutated at smaller frequencies, it's called hills, that also are important in the landscape. So for the first time, knowing not only that there are these important genes mutated at high frequencies, but also these sort of lesser players, but also contributing significantly to cancer, but really showing that the complexity of cancer and really sort of launching the field of cancer genomics in that sense. Subsequent studies was done for pancreatic cancer, and not only looking them no longer as single genes, but be able to group those genes as pathways. So even though that specific tumors could have different genes that are mutated, but they share common pathways between the different tumors. So that for pancreatic cancer, we were able to identify approximately 12 pathways, of which even the different tumors, although they have different individual genes that are mutated at different pathways, that they share the common pathways between the different specific tumors. And subsequently, at the same time, we were also able to perform the genomic analysis for brain cancer, specifically for GBMs, and we're able to discover a very important new gene, specifically IDH1, which is shown in the figure here, a well-known metabolic gene, but mutated at a very high frequency and actually clustered around a very significant active site. And what was important about this, not only was it able to be shown to be mutated at a high frequency, it actually defined a specific subtype of cancers. People with gliomas who have mutations in IDH1 actually perform and have a better prognosis than those who do not. Not because that specific mutation, but that this defines a new subtype of gliomas that have sort of IDH1 mutations. And currently, there are many people working on specific therapeutics based on IDH1, and even within the last few weeks, there have been very promising results have been reported in mice using IDH1 inhibitors to be able to show effectiveness in treating glioblastomas. So with all these sort of initial findings in cancer genomics, starting from single genes, looking at gene families, and eventually looking at genome-wide in terms of the exome, this really then allows us to start thinking about how do we implement this on a clinical scale and be able to really impact patients in that way. And some of this work was done by colleagues and I at Washington University in St. Louis, where we were starting to take some of these initial research findings that was done in a research environment and start thinking about how to implement this on a clinical environment. So done in a new space, specifically dedicated to this within the pathology department, how to implement not only sort of cancer genomics, but doing it in a clinical context. So the first important thing is this wasn't done within a vacuum. it was important that it was a part of already existing sort of pathology services. Had a really rich in terms of faculty, over 150 faculty, and informatics background with significant research background, and combining technology expertise, informatics expertise, and pathology expertise to be able to bring this forth. And importantly, to be able to understand this, how it's different to do it within a clinical context versus in a research context. So this is, and sort of the difference is actually significant and very sort of non-trivial, how clinical sequencing is different from research sequencing. So one of the first things we thought was we actually designed a gene panel to be able to do. We also later did it in terms of exome. And then how do you select the genes if you want to design your own gene panel? And for us, at WashU, we had sort of 4 criteria. Instead of just sort of sequencing, tens of thousands of genes, we wanted to choose the genes that were of clinical importance. So the first criteria was clinically actionable. So if you were sequencing genes and if the results that you receive, you can't sort of act upon it, it is hard for it to be a true sort of clinical test. So clinical actionability was a very important factor in terms of the design. Secondly, it may not have a specific drug sort of tied to it, but it also could be prognostically relevant in terms of important in terms of deciding whether this is gonna the course of the patient, whether you want to treat more aggressively or not. So that was also very important. The third aspect was in terms of reimbursable. This is very sort of practical aspect as we design a clinical test that the payers will be able to reimburse it. So often these would be genes that are already reimbursed through Sanger sequencing, and we would be using a different modality to be able to use it, to be able to sort of test for these genes. So this required a lot of sort of conversations with payers to be able to determine what genes would be reimbursable. I think with larger panels, with a lot of these genes that are more sort of research level, it makes it harder to be reimbursable as a clinical test. It could be useful sort of as a clinical study, for truly be a reimbursable clinical test. The curation of these genes was highly important. And the last aspect was a gene set that the physicians clinically already use it. So because these are the people that would be ordering these tests, it was important for them to have the gene panels that they already understand, because the education part was going to be very significant. So including genes that they already understand is very, very important as we sort of design these panels for them, so that when they look at the panel, they already include the genes that they normally use, in addition of genes that they potentially will consider adding onto the panel. So these were sort of 4 very significant aspects in terms of thinking about the design. So of the specific genes that we initially started, there were sort of complement around 30 genes that we looked at that included both importance for solid tumors as well as hematologic tumors as well, as you can see here, and some of them are for both. And as you can see here, in IDH1, IDH2 are genes that we helped discover were also part of this panel and of great importance. There's some that are sort of highlighted in bold that have specific targeted therapy with it, for example, or of higher importance, such as EFGR, known for lung, and different targeting cancers. So this was sort of the gene set that we used for that to be able to, and in terms of our curation of that initial set. In terms of the workflow, we show here quickly. The standard workflow is very similar. So from steps 1 to 8 here, that's shown in this diagram, it's very much shared between the standard research workflow and the clinical workflow. So whether it's a fragmentation of DNA, preparing the library, enriching, and then doing the standard bioinformatics of alignment and band files is all the same between research as well as clinical. But what was very different then is from steps 9, 10, and 11. So step 9 was very, very important in terms of sort of the tier 2 informatics. Specifically, in a research context, if you sequence and have a result, you can just report the result right in the paper and it's done. But within a clinical context, these results are acted upon. So high confidence about these results was going to be very, very important. And this confidence of the results is not only done on a gene level, but it has to be done on a mutation by mutation level. So here for us, in order to build, be able to interpret this, there was a lot of work done to build a clinical grade database to be able to curate for not only every single gene that's in the panel, but also every single position in the panel. So that every of these positions, as we generate these reports, that the confidence is not done on a gene level, but on a mutation by mutation level. And there's different levels of confidence you want to build for this. For example, the highest level of confidence, of course, if you have evidence of that specific tumor type in a specific gene in a specific position with human clinical data to report, that's of the highest confidence. And that level, sort of the highest level of confidence we would report out. However, not all mutations for all tumor types have that level of confidence. So a second level of confidence would be then if you have a specific mutation of that gene in that position, but not necessarily of that tumor type. Right? So for example, you have an EGFR mutation, not specifically in lung, and a different tumor type, and that could be a different interpretation. A third level of evidence that can be sort of presented is it's shown in that gene, in that position, and potentially in that tumor type or not, but it's not specific human clinical data. So whether it's an in vivo model or in vitro model, those are sort of more experimental models and not necessarily clinical, and that's sort of a level of evidence down. And you can sort of surmise in terms of the different levels of evidence as we go down is, not necessarily level 4 would be not necessarily in human, but in animal models, which is shown. And then level 5 and further down would be not even in animal models, but in sort of bioinformatics in silico prediction models. So this level of informatics is really a huge difference between what one does in a research context versus what's done in a clinical context. Step 10 is also very significantly different in terms of clinical genomics versus just research genomics. Is that there's a clinician that ultimately signs out. Often for us is we have a specific pathologist who understands not only the biology and medical aspects, but also the informatics and research aspects. That's able to take all the information that we're able to convey from our databases, from the raw data, and to be able to then craft a report to be able to do that. And this is not an insignificant step. It often takes many hours to go through all the literature and to be able to sort of create the best, curate all the best evidence and be able to sign out the case so it could be helpful for oncologists. And finally, step number 11, then the report for the oncologist would be one that's able to summarize all the data and one that is understandable from oncologists without significant sort of training, with even understanding NGS, that they can be able to, so that the average oncologist would be able to understand this data and potentially, for example, point to existing clinical trials, point to targeted therapies that might be helpful in these aspects, and really providing all the facts that's going to be helpful in clinical decision making. So some of the sort of the technical challenges we want to think about in clinical context versus research context are a few. I list some here. For example, working whether with FFPE or frozen samples versus sort of tissue lines in a research context. Often another is the amount of specimen that you have is very small, whether from a biopsy or cytology. So that's something to be able to do. Number 3 is be able to detect variants as low as 20%. Right? So tumor cellularity, you may not be able to, from your specimen have high tumor cellularity. So the variant detection in this context would be very important. Microdissection, sometimes not necessarily is beneficial, but be able to do that because microdissection takes significant amounts of time. Read depth because of tumor heterogeneity, we usually sort of look at between 500 and 1,000 fold. And then, often in order to reduce costs, a significant sort of multiplexing And I think, as previously said, that the report must be simplified so that it is not one that's needed for sort of next generation sequencing experts, but for the average oncologist, and then the clinical grade database is going to be important in terms of the difference between a clinical sequencing versus research sequencing. So this is a graph of our sort of sensitivity in terms of being able to detect in terms of 10%. And you can see here shown in the black is the sensitivity, and then the red is the positive predictive value. So as you can see, on the x-axis is the coverage, and the y-axis is percentage. There's a really sort of quick increase in terms of sensitivity in the early with coverage, but there's an inflection point probably around the sort of 1000x, where you're able to detect about sort of 90% percentage at sort of 10% allele frequency. And this is going to be important because of heterogeneity in tumors for this aspect. And then finally, this is sort of the list of the genes that we use in our sort of smaller panel. In combination, we use in terms of FISH and Sanger as well as NGS, and then the different tumors that it's in. Different tumors obviously have indications of which of the genes that will be more helpful, but because of cost associated, sometimes it's just as cost effective, it's just to do the whole panel. But for example, the specific targeted therapies are to specific tissue types, and those will be important. So whether it's solid tumors or liquid tumors, the specific genes would be important for those aspects, and this is shown here. So the oncologist has the flexibility of ordering just for example, the AML panel or the lung panel or the melanoma panel, or the option of ordering the entire panel for their purposes. So this is sort of for our first time, really. I think we're excited that patients are being helped on a daily basis, not only from the research aspects, but from a clinical aspect from as well. So for sort of the next half is we're going to sort of specifically talk about what are some of the lessons that can be sort of taken from our research aspects. First of all, in terms of thinking about participant selection. So you see here on the x-axis is whether the decision-making is done on a group or on an individual level, and then on the y-axis, whether it's done on clinical versus research. And the traditional is starting from the quadrant on the bottom left, where in a research context, a group makes a decision, and it's done in a research context. But ultimately, then the movement is to establishing another clinical text that is in the top right corner, where an individual is able to make the decision, and it's done in a clinical context. So two specific sort of challenges must be done is, first of all, criteria development to be able to move from a group decision to an individual decision aspect. So who are the people you're going to be ordering this test on? Whether it's people with refractory cancer, or you can even would you be able to order this test actually pretty early on. So this criteria development is able to then no longer rely on a group to be able to done on an individual. From moving from research to clinical then is sort of shown here as sort of clinical test development, where establishment of, for example, the clinical grade database, having clinicians be able to sign off the reports, creation of the report that's sort of clinically interpretable is going to be important. So ultimately, starting from the bottom left, moving from the top right, is sort of the implementation of our test. In terms of the study design, in terms of the coverage, a lot of people have sort of talked about, in terms of the number of genes that one should do, which is sort of shown here on the x-axis versus sort of the depth of coverage that you'll get. Obviously, with a finite number of reads, there's sort of a trade-off between breadth versus depth. And traditionally, if you look at the far left, right, single gene Sanger, you're able to sort of sequence at a very high depth. So the trade-off here then is whether one does sort of a gene panel versus exome versus genome for cancer. For us, we think because sort of with data shown earlier, that the coverage, because of tumor heterogeneity, if you want to detect stuff, mutations at 10%, it would require actually very significant coverage, potentially even 1,000x, 500 to 1,000x. So shown here in purple is sort of a gene panel with cancer. This allows you to be able to sort of catch the genes that are heterogeneous, the heterogeneity of these cancers, even at low amounts. However, you can, people obviously haven't been able to create, a test in terms of a whole exome, but the coverage definitely would not be as significant. So you will miss, be able to sort of coverage in terms of the heterogeneity in that aspect there. And ultimately, people also be able to work on whole genome, but for cancer, really, it's hard unless you're able to significantly, not think about the heterogeneity. So whether you have a lot of the specific specimen, or whether you're able to sort of grow that specimen in terms of a cell line to be able to do that. But in terms of our understanding and working with patients, we really think that a gene panel, not even a large gene panel, a smaller gene panel that's highly curated, that's highly actionable, that's highly informative, and be able to cover that at depth is potentially sort of a better approach. The one that you want to do sort of a significant number of genes, of the findings you find you don't know what to do with. So that's a big difference between sort of clinical and research. And then we would think then, if the gene panel is negative, then people are interested in doing more deeper sort of analysis. Then the whole exome approach can be done, understanding that, of course, the coverage will be less and not covering in terms of the heterogeneity. In terms of the sample prep, in terms of how to be able to sort of enrich for these areas, of course, there are sort of two main aspects that people do, whether you want to do the amplicon approach in terms of enrichment versus the capture approach. The benefits of the sort of amplicon approaches use is that you can be very sort of specific in terms of what you want to amplify out because of your primer design. And this is done in panels, which is shown here, whether it's the AmpliSeq or using Fluidigm or RainDance systems for panels. Or you can use capture system. The capture system is, you're able to do a lot more often. And the exome approach for capture really has been the dominant approach here, whether with NimbleGen or TrueSeq or Agilent. Or with sort of smaller panels at different institutions, be able to see. So whether, I just list some examples here that are able to, build their own panels and capture. The benefit of this is actually it's pretty, simple to sort of design a capture panel there. The downside of sort of amplicon systems is you will lose the information from copy number from that, if that's something of interest there, because of different amplification. So you would lose that information. So that would allow really think that capture methods are a better sort of method there. So sample prep. In terms of the technology, I think we've seen the come and go of many different second-generation technologies, and whether it be Solexa now being Illumina, or Life initially purchasing ABI and now using iTorrent, and potentially with and then technology that are no longer as popular, whether it's Helicose or 450, or complete genomics. But obviously, everybody is looking towards the third-generation technologies. Obviously, most excited with people with Oxford Nanopore, and with the new system, the new bio now announcing their system, or whether we're Nabsys. But the importance is here is that as we talk about clinical sequencing, the important aspect actually does not lie within the technology. The important aspect actually lies within annotation. Lies in the gene curation, lies with the level of evidence that you provide there, lies with the personnel that you use to be able to sort of sign out these samples, so that even if these different technologies are rolled online, what's important in terms of the clinical aspect is that database that's used for annotation for that. So I think third generation will be able to obviously reduce in cost, and be able to potentially be able to look at heterogeneity better. But the interpretation systems ultimately still is going to be most important in terms of the bioinformatics. So since we're talking about informatics, there's a sort of rough outline, whether from the reads that you get from the machine, you start sort of doing alignment, doing variant calling, alignment, most people, whether BWA, or bow tie or SOAP, and then variant calling, GATK, SAM tools, and then be able to then filter based on what. So there's a movement whether people sort of do it in terms of locally or cloud there, and I think and people have sort of different options there. It seems like a lot of people now have been able to sort of do most of the alignment and variant calling in a local aspect, which is shown in the orange, and then some of because the annotation is sort of built in the cloud, will go in the green. So shown in the orange and the green is often implemented in many systems. Be able to then leverage annotation that's done in the cloud. However, there are systems where that people are able to do alignment and variant calling in the cloud, but because of the large file sizes, to be able to do that, actually much easier to do that locally. And then you see the purple here, then there's people with solutions be able to then do not only the alignment variant calling, also the annotation locally or in the call as a system, but then rely on a clinical reporting from a third source. And ultimately shown here at the very end is whether there's the whole system done locally in the cloud. So ultimately, clinical genomics and for cancer can be done either in really a sort of a distributed system where everybody has their own MySeq or torrent and able to do everything locally and do that, or you can think of it where it could be a centralized system, such that one that Complete Genomics was hoping to point on. That it's done all at a large site where samples are sent. So this is still to be yet determined whether clinical cancer genomics will be done in a distributed manner from all local labs running sort of PGMs and MySeqs, or whether it be done in large genome centers like the Broad or BGI in that context. And this has, clinical precedents. For example, in the U.S., clinical testing, whether done by big companies like Quest or LabCorp, or whether local testing at hospitals as well. So both are models that are considered, and the informatics follows that very closely. And lastly, as you think about the informatics, how to then, interpret it not only in terms of matching known variants, but looking at population frequencies, looking at known annotations, looking at the genetics, but also using prediction algorithms, whether it be ones that are very well known like SIFT and Polyphen, or many of the ones that are coming out that are able to take into consideration homology modeling, 3D structure, functional annotation as such, to be able to do that for filtering. And in terms of the reporting, this is a very, very area of active discussion is what do you then report back out to the patient? And I show here quickly, in a simplified manner, potentially there are two axes, going from on the x-axis from a closed, you don't reveal anything to the patients, to an open platform, you can tell everything, and then on the y-axis from research to clinical. So traditionally has been done sort of a closed research system, which is shown in the bottom left quadrant. And then as more and more sequencing is done, there's discussion of a reporting of incidental findings and how that is important, and a movement of more doing, to the top right quadrant where it's done on a clinical aspect and a more open basis. And here I show some examples. The most extreme here, obviously, being the Personal Genome Project, where individuals are able to sort of publish their genomes online through very, very rigorous consent procedures. But the movement here within the field is going to be more and more open. Of course, they're going to be requiring a lot of sort of public education to be able to sort of give all that data back, whether it be exome data, whether it be specific gene panels. But that's sort of the movement you can see here as research goes to clinical from closed to open. And then I think ultimately, I think like other sort of specific testing that we really, really shouldn't think about what people call sort of genetic exceptionalism, where we think genetic data is different from any other clinical data. We need to protect patients from it, but more, we can give them access, whether people are having their whole genome sequence when they're born, and then be able to reference that as they use it later on in life. And reimbursement often is also very key as well, and I show this in terms of as a rough, shown in a log scale in terms of cost. And NGS sequencing is shown here as a second bar here, whether it's sort of exome genome between sort of the $1,000 to $10,000 level, but which is very actually considered, in the same range as whether it's BRCA testing or scale gene testing. So in terms of cost effectiveness, is similar there, and that will obviously continue to drop, and which is very comparable, obviously, to MRI, CT scan, colonoscopy, or sort of extended hospital stay. So I think we think reimbursement is as the prices drop, this is not going to be a significant aspect if it's going to be justified in terms of clinical decision making. So at the end here, it's important to say that whether it be panels or genomes or exomes, these are not perfect systems. So I'm going to sort of walk through some of the caveats that you want to think of. Number one is insufficient power annotation. I think we talked about the clinical database, but most of the variants that you'll be able to discover potentially are of unknown significance. So that's going to be important. Secondly, even if you do exome, there's what people discuss about sort of first exon drop off in terms of high GC coverage, and the coverage is going to be a big aspect, especially with larger panels. That even if you cover at a 90%, that's a significant amount of genes and exons you're not covering. So it's important to differentiate lack of coverage versus not finding it. A gene in that aspect. So that first exon problem continues to be one of great significance. Next, I think that in terms of the informatics, the field is better at looking at single nucleotide variations, but there's a new method now looking at copy number alterations and translocations, but that still is sort of within the works to be able to do that in a rigorous manner. So right now, most of the emphasis has been on sort of single nucleotide mutations, but as technologies mature, we'll be able to look at all these different genetic alterations as well. And even as we do this, even if we do exon, of course, we miss out on the introns or microRNA. So just because we don't sort of see it in our panels doesn't mean that it doesn't exist. So that's going to be important. And I think the bottom ones are the two are sort of specific about sort of Mendelian diseases, and this is sort of less important within sort of cancer, but they're going to be sort of non-genetic or non-Mendelian type of. So whether in the second panel here, thinking about epigenetics or microbiome, and specifically with cancer, you want to think about whether you're looking at somatic mutations where you're sequencing both the cancer as well as the normal, or you're just sequencing just the cancer. And there's different pros and cons. And obviously, you'll get better results by sequencing both the cancer as well as the normal, but again, there are sort of cost considerations. And lastly, sort of in terms of alignment and variant calling, although the algorithms are pretty mature in themselves, but even if you run very sort of well-known algorithms, there are very much differences. Even if these differences are, let's say, even if at 95 concordance rate between these different algorithms, that 5%, if it's sort of the right 5% that you, in a clinical setting, that is very, very significant. If 5 out of 100 of your patients have a wrong call, that's going to be a very much a big problem there. So I think the alignment algorithms, the variant calling algorithms, as well as the annotation databases, we must understand that they're not 100% right now. And using that in a clinical context, whether for precision medicine or whether for tailored therapy, you need to think of that as well. So that's just sort of in brief, quickly, in terms of sort of our experience looking at cancer genomics. And the excitement is how all the research now is now being translated, and we're really, really excited that patients are going to be specifically be helped. And so we're very there's obviously a lot of people to thank, but these are just some of the people that have really sort of contributed in terms of our work. And then if you have any sort of questions, this is sort of my email, and feel free to connect me from there. So good. So a little bit of time for questions and answers, which I will start taking a look at. Oh, and for those who want to get continuing education credits, this is the link to get specific credits there. I'm going to leave it on the emails for those. So I'm going to now answer some of the questions. So the first question we have here is from University of Massachusetts. Dr. Van, thinking about what is the biggest obstacle to demonstrating the value proposition of molecular diagnostics and increasing reimbursement for these tests? That is a great question in terms of what is the clinical value. So it's a couple of things, and this is really built into how we select our gene panel. First of all, you need to show that it does affect clinical decision making. So if your panel really consists of genes where even if you find a mutation, it really doesn't affect your care of the patient, that's not going to be very convincing to payers. So it has to be genes where, for example, like EGFR, where there's sort of targeted therapies where you can really make a difference, and clinical decision making is going to be very, very important there. In that same sense, actually, the contrary also is going to be important. Is that there's not going to be stuff that's of less sort of helpful to clinical decision making, meaning panels with large numbers of genes, of which many of them do not meet those criteria. Then the payers can just say that this looks much more like a research panel, because if most of these genes, even if you find a mutation, you really won't be able to affect the care. So there are thus then possibilities for them to be able to argue that this is not a clinical test, but in terms of just more of a research test. So the demonstration of the value really is sort of clinical value, and if you're able to sort of show that this really changes how you treat patients, that is truly the ultimate metric to be able to do that. Don't see any other questions coming. So if people have questions later, feel free to email me. If not, I guess we'll be able to then end this session early then. Thank you very much for your time and attending this conference. And feel free to email me if you have any questions. And thank you for BioConference and the conference organizers for putting this together. Thank you.